NGS is the most indispensable component in the precision medicine infrastructure because it provides genomic ground truth.
The actual instruments that decode human genomes are evolving very rapidly. They're becoming faster, they're becoming more effective, more accurate, they're becoming less expensive.
Do I see this as a world where everyone will get their genome sequenced? Absolutely. Probably before they're born, in fact.
Next-generation sequencing, or NGS for short, is a powerful tool that allows for rapid, large-scale, and cost-effective analysis of genetic material. This technology has advanced the way scientists investigate genomes, transcriptomes, and epigenomes, leading to quicker and more accurate disease diagnosis and the identification of potential treatments. In this segment, we will explore how NGS has become a routine component of the precision medicine workflow. Offering personalized treatments and reshaping our approach to healthcare, sequencing has significantly improved numerous fields like oncology, infectious disease monitoring, rare disease diagnostics, and more.
Human genetics defines how our bodies react to disease and treatment. Precision medicine means using that information, which is specific to each individual, to guide care.
We use next-generation sequencing for sequencing people's genomes and predicting risk. We also measure it for measuring the outputs of genome, meaning your RNA, which gives you some indication of what genes are expressed and also a clue on your health as well. And likewise, we actually measure your microbiome with next-generation sequencing, which is very valuable as well.
Next-generation sequencing, or NGS, reads multiple genes at once, and that can help us identify disease-associated variants to help match patients with the most effective therapies. It could also help assess disease risk, develop more targeted therapies, and reduce healthcare costs overall. In other words, precision medicine can enable your doctor to tailor treatments specifically to you, selecting the most appropriate therapies for your unique genetics.
I think the dream going way back to the 1990s when people started the Human Genome Project has always been, can we do this routinely? Can we make this part and parcel of your medical record so that you will have this at your doctor's fingertips so that they can predict what diseases you may develop, and so that we can take preventative steps. And the moment symptoms appear, your doctor will know what it is, as opposed to the way medicine's practiced today, which is very reactive.
We spent $3 billion plus analyzing the first complete human genome sequence from many different kinds of genomes. And the first sequence roughly cost $1 billion to do. And what the Genome Initiative did was set in place the technology development for improving sequence so that today we have 7 orders of magnitude less cost. You can begin to do a genome for almost $100 a genome. And from $1 billion to $100 is a striking change.
While sequencing costs have dropped dramatically, data analysis still requires expertise and computational resources, which add to the costs. Yet overall, sequencing has become more affordable and accessible for precision medicine.
NGS had an enormous impact on precision care, starting with fundamental discovery research. In there, sequencing helps illustrate the molecular drivers for both good health and disease. As sequencing moved into the clinic, it has provided enormous insights to guide care.
Due to the wide range of available sequencing systems, each with unique underlying chemistry, we will focus on one of the more well-established systems from Illumina. This process begins with isolating the nucleic acid from the patient samples. If the patient transcriptome is to be analyzed, the RNA must be harvested and converted into complementary DNA. Next, DNA strands are separated and fragmented. Adapters are then attached to both ends of each fragment. These adapters contain sequences essential for sequencing. The samples are then loaded onto a flow cell. Each DNA strand randomly binds to complementary adapters on the flow cell's surface, initiating cluster generation. During cluster generation, DNA strands are amplified through bridge amplification, forming densely packed groups called clusters. Once the clusters are formed, the samples are ready for sequencing. In the sequencing phase, fluorescently labeled nucleotides are incorporated into the DNA strands base by base. This process continues until the read length or the desired sequence length is completed. An optical system records the fluorescence emitted as each nucleotide is added, determining the sequence based on the color detected. This process continues depending on the read or chosen length of the sequence. Finally, data analysis involves sorting sequencing reads by sample, grouping similar sequences, and aligning them to a reference genome. This helps identify variations or mutations, providing biological insights or clinical information. In recent years, next-generation sequencing has transformed our understanding of a number of different diseases, leading to improved methods for diagnosing and treating them. The progress in NGS technology has made it possible to perform detailed genetic analyses with minimal tissue requirements, resulting in its increased use in precision medicine. As sequencing offers faster results and more personalized patient assessments compared to traditional methods, the adoption of NGS is expected to continue growing.
The other area where genome sequence has been super powerful is for cancer. So cancer is a genetic disease. You can either be predisposed to cancer, like the BRCA mutations, or it can come up what's called somatically. As your cells grow and divide, mutations pop up, and that can lead to cancer.
Investigating tumor genetics can reveal the molecular variations that may be driving rampant cell growth or allowing the cancer to resist treatment. On a research level, these potential therapeutic targets flow into biopharma companies, which then develop small molecule compounds, antibodies, and any other agents to modulate those targets. On the clinical level, NGS can read each patient's tumor genomics, matching their cancers with the most effective therapies. NGS has been and it is particularly powerful because it can answer many questions about a patient's cancer in a single test, providing far more information than traditional pathology, which often answers one question at a time.
In one study we ran, we sequenced 70 people, and 12 of them had learned something pretty important from their genome. Someone had a mutation in a gene that put them at high risk for cancer. They did a whole body MRI scan to see if they had any, they're called lesions, what have you. And it turns out they actually had early thyroid cancer, which was caught because they did a follow-up to their genome sequence. They had that part removed. They were able to keep most of their thyroid. And the net result is they don't need thyroid replacement therapy or anything like that. Again, it was all caught because we sequenced their genome.
NGS is an essential part of this equation. Without the deep genetic insights sequencing provides, it would be impossible to know the genetic drivers underpinning a patient's cancer or even begin to counteract them. NGS can also show a person's hereditary predisposition for specific cancers. So knowing that a patient is a higher risk, physicians can develop a personalized screening regime.
This will be the future where with next generation sequencing, you'll actually be able to detect early cancer in people's blood.
Next generation sequencing has also significantly advanced rare disease research and treatment. Its rapid and precise genetic analysis has been especially beneficial for diagnosing and treating pediatric patients with rare conditions.
Rady Children's Institute for Genomic Medicine is a research institute, and we are focused very much on delivery of precision medicine to children, hence our name. And we do that by decoding genomes.
About 4% of kids are born each year who have a disease that's not clear what's going on. Half the cases they figure out with standard tests, but the other half they actually can't figure out. So people have turned to genome sequencing to figure out what's wrong with their child.
We have about 4 or 5 really big programs that we are focused on. One is decoding the genome in critically ill children. These are children in intensive care units at now 100 different children's hospitals around the country. And each day we will get samples from patients together with their medical record. And it's our goal to identify whether or not they have a genetic disease, and to do that in a day. That's tough. We do this about 1,200 times a year, and that's our single biggest project.
Sequencing has also had a remarkable impact on the rare and undiagnosed genetic disease community. Prior to NGS, patients would receive single gene diagnostic tests. Based on the patient's symptoms, clinicians will then guess their disease and test for genetic variation they believe was causing it. However, there are more than 7,000 rare diseases and many share symptoms. As a result, the clinicians would often be wrong. The single gene test would come back negative and a new test would be ordered. This process could be repeated dozens of times over many, many years.
So we are all about changing outcomes for children. Initially, it was about getting a diagnosis. There's this thing called the diagnostic odyssey. On average, children wait 5 years to get a diagnosis of a rare genetic disease. And so we wanted to collapse that down to a day. Could we diagnose in a day? And we can. But the next question is, how can we start effective treatment in a day? Because if all you do is label the child's illness, you have not actually saved their life. You've not alleviated their suffering. So that has been what we've really been focusing heavily on over the last 5 years, is how do we make sure that every child we diagnose gets the very best therapy.
One of next-generation sequencing's greatest strengths is its comprehensiveness. Patients with rare diseases, cancer, or other genetic conditions often face a diagnostic odyssey, receiving negative test after negative test. NGS can provide validated answers, rapidly allowing patients and clinicians to pursue precision care.
One of the things that unites the folk I work with is this desire to impact individual children's lives and individual families. And so we share that information. And each week, it's our goal to say, did we help a child this week? Is there a story in terms of a child whose outcome was better because of what we did? And we count those, and we measure them, and we get pictures of those children, we frame them, and we put them on the walls around the Institute. And each year we produce a book that outlines some of those success stories, and we share that with people who are interested in what we do. So these are the stories that make what we do meaningful. It's what we value as an institute. It's not so much whether or not we publish papers in high-ranked journals or whether we get famous. It's much more these stories about families who've gone from the worst day in their life to having hope.
This dedication to making a real difference drives the continuous innovation in genomics. Over the years, advances in next-generation sequencing have been essential in making such impactful work possible. These technological developments have transformed sequencing into a core component of the precision medicine workflow, enabling more accurate diagnoses and personalized treatments.
I think the difference between then and now is that now, first of all, technology has advanced quite a bit. It's more accurate. People are more comfortable with how we interpret genomes, so the world has warmed up a little bit. But I also think we can add other kinds of data too, and that's turned out to be very, very powerful, meaning you don't just get a genome sequence anymore, you can get additional information that can also be useful for helping make predictions about people's risk for diseases and things like that. And so I think the field is emerging very, very rapidly in many different directions.
I think another technological advance is this enormous reduction in cost and speed of automated DNA sequencing.
Genome sequencing has gone from being pretty crude to now much more accurate and pretty inexpensive. Pretty soon it's going to be affordable to everyone. And I can envision a world where everyone does get their genome sequenced to better manage their health.
Along with the drop in costs and rise in accuracy, scientists have developed new techniques that have further advanced scientific discoveries. In particular, single-cell sequencing was a major breakthrough that has improved our understanding of cellular behavior and interactions. This technique allows researchers to analyze individual cells in unprecedented detail, providing insights into how cells function, communicate, and contribute to health and disease.
Advances in single-cell studies had led to spatial omics, in which researchers study how cells interact within their biological context, their neighborhood. NGS is the root technology for so many advances: synthetic DNA, CRISPR-Cas9 gene editing, single-cell studies, and so much more. Each innovation gives us a new window into biology and greater opportunities to understand ourselves and help people.
We started precision medicine by decoding the human genome, which is 3.2 billion letters. It's a book that would be 400 feet tall if you printed it. But now we are adding to that other omic technologies. For example, the ability to measure 5,000 different proteins in a drop of blood, or using AI with the electronic medical record. Some children's medical record is thousands of pages long. Can we use AI to just suck all the information out of that?
Using deep learning to decipher genetic data is emerging as a crucial innovation in genomics. A recent study led by researchers at the Illumina Artificial Intelligence Laboratory and published in the journal Science showed how these advanced algorithms can help identify disease-causing gene variants to diagnose rare diseases. The technology, called Primate AI 3D, uses deep neural networks, similar to ChatGPT, that have been trained with human genomic data. These innovations mean we can do more with this sequencing information. Variants that previously may have had uncertain significance can now be rapidly clarified through AI and other computational technologies. In other words, it will provide more definitive answers that will directly translate into personalized care.
By combining these technologies, we make each of them much more effective and have much broader impact. And that really is a future direction where we have a lot more yet to do. There's a lot of new technologies that are genome-like in terms of they're comprehensive. They measure everything at once, but it's not DNA. It might be biochemistry, measuring every chemical molecule at once in a human blood sample.
These advances are just the beginning. With continuous developments and the integration of other innovative technologies, next-generation sequencing holds immense potential for transforming healthcare and improving lives.
There is so much more on the way. In a few years, new and even more mind-blowing technologies will emerge, all enabled by genomic sequencing.
Stay tuned for our next segment, where we'll cover how spatial biology is advancing precision medicine.